Good morning. I'm delighted to be speaking at the Cell meeting on the Med. I'd like to thank previous speakers for being efficient with their time, and we'll try to get us out to coffee at the appropriate time. I'd like to introduce you to our cell manufacturing platform, which is called Solupor. And during the course of the talk, I'd like to speak to three topics: cell manufacturing, cell health, and cell functionality, and specifically with reference to two key applications: iPSC manufacture and T-cell manufacturing, or T-cell therapeutic manufacturing, and speaking specifically to the health and function of cells for more potent therapies for the benefit of patients. So these are two embodiments of our platform, Solupor. On the left, we have our research tool, which is a compact, easy-to-adopt embodiment of the technology. It's highly reproducible. We've tech transferred it on several occasions to partners, and it's our main tool of adoption. On the right-hand side, we have our automated, closed clinical system for commercial manufacture. This is called SUS, or single-use system. We have established a GMP supply chain for the clinical device in the US and are positioned to support partners with regulatory filings in the back end of 2024. I think what's critical here is that we've both tech transferred both platforms to partners, and that there's translatability between them, so the initial preclinical work done on one platform will translate onto the other. So a word about the mechanism of action. So what is Solupor? So Solupor is a novel, robust physicochemical transfection process. It's not electroporation. There's no electrical aspect to it. There's no virus or particle or lipid. It's a cell membrane permeabilization technology. So what happens is cells are introduced into the chamber on the left-hand side of the slide, and when they're introduced, they're taken out of suspension onto a highly engineered filter at the base of that transfection chamber. When they're taken out of suspension, they're never desiccated or dried, just the excess media taken out from on top. They are precision sprayed with an atomized solution of our permeabilization chemistry, a proprietary permeabilization reagent, which is indicated in the vials on the top there. And three things happen. First of all, upon contact with the cell membrane, the solution of membrane permeabilization reagent and the cargo or cargoes of interest immediately disrupt or fluidize the cell membrane at the same time as the cargo or cargoes are concentrated at the membrane surface, and the atonic gradient is set up from the outside to the inside of the cell. The mechanism of transport is passive diffusion, and immediately that this happens, and it's a very rapid process, the cells are resuspended in the media through the base of the device, and they go on to the next part of the manufacturing process. We, at this point, have conducted, both in our own hands and the hands of partners, thousands, if not tens of thousands of transfections over multiple donors, primary cell type cell lines, and with multiple cargoes and combinations of cargoes. So it's a very robust and consistent process, and it's extremely well tolerated by cells. And that's a good segue into the second topic of cell health, which is central to our value offering. So I think this slide indicates one way that we can measure or look at cell health is the perturbation of cells which are modified. And in this, on the left-hand side of this data, we're looking at a nanostring panel of about 600 or 700 immune-indicated genes when looking at products for immuno-oncology, for instance. And what we see is we're electroporating a population of cells and Solupor modifying a population of cells. And we're looking at the delivery process only here. And what we see in the electroporation population is growth up and down regulation of gene pathways in the cell in the face of the electroporation process, and comparing that with the solupore process underneath, which is a very much quieter. There isn't that same gross up and down regulation of factors. We see this at the 6-hour time point and then at the 24-hour time point. And although the picture does settle down at 24 hours, in typical gene editing processes, the gene editing is completed at that stage. And what are the gene pathways which are up and down regulated? They're the ones which should be of interest to us concerning the health and function. They are the pathways of activation, metabolism, exhaustion, and so forth, so of keen interest to therapeutic developers. So I spoke to a number of applications for using this technology, and the first application I'm going to speak to is iPSC manufacture. And in this data here, we're looking at the enabling technology for reprogramming of iPSCs, and specifically transient reprogramming, where we're not using Sendai virus, for instance. So this is to go from a CD34 somatic cell to deliver to it for the purposes of reprogramming it. And here we're transiently delivering mRNA factors with high efficiency and high viability of the cells. And then when we look on the right-hand side at the proliferation, the expansion of those cells, it's comparable between the untreated control and the solupore modified cells. So with those iPS cells with those cells now transferred into or brought into an iPS state, the second application is editing in the iPS state. So once again, using mRNA delivery on the left-hand side, we're looking at high delivery efficiency and viability of the cells which are being edited. And on the right-hand side, we're looking at RNP editing of the iPS cells. And once again, through a sequence of edits, so repeat editing of these cells, we're seeing high viability and delivery of a knockout gene here. So laying these over a typical workflow, so putting it all together, if you like, starting with CD34 progenitor cells on the left, we can follow any of the pathways in this workflow where we're reprogramming, editing, or guiding, differentiating those iPS towards their terminal cell type. And in so doing, avoiding the cost and the footprint, if you like, of using Sendai virus. So this is of keen interest to the iPS manufacturing industry, and would be delighted to pick up the discussion with anybody who is interested in that in the room today. So switching gears here and speaking to the second application, which is T-cell manufacturing. We're looking in this slide at 3 panels here. First of all, knocking out the track locus in human primary T cells, knocking in a marker GFP integration gene with high efficiency, and then overall through that knockout and knock-in, looking at the viability over 2 donors here. So high viability, high efficiency knockout, and relatively high efficiency for what's typical in terms of integrative knock-in, and that integration measured out to 7 days on this slide, and in our own hands out a lot further than that. So moving on from that knockout, knock-in, one of the key pieces of work we did last year was the development of a model CAR T, a complex CAR T product, which includes a CD19, a lentivirally transduced CD19, along with 3 knockouts, CD7, B2M, and TRAC. And in this multi-donor study, what we're doing on the left-hand side is we're showing, we introduce a comparator here, which is electroporation. And what we're showing is the comparison of the population who have those edits introduced by solupore versus those that have the edits introduced using electroporation. The electroporation is in the purple or blue here. So what we can see is in the left-hand side, greater preservation of the stem central memory subpopulation in that population of triple edited CD19 cells with the Solupor manufacturing step. And on the right-hand side, when we look at secretion of interferon gamma from this population of cells, we see as a marker of cytotoxic activity, we see that the interferon gamma is higher coming from these cells which have been Solupor modified relative to those which have been electroporated. And then when we look directly, specifically at cytotox, the green line here is indicating the same population of cells being more cytotoxic than the cells, the population which have been engineered or manufactured using electroporation. So on the back of that in vitro work, we then did a study at Jackson Labs at the end of last year and the beginning of this year, where we introduced that complex CAR T product into a population of mice. And you see on the left-hand side here, the only difference in the two populations is how those triple edits are introduced that I spoke to in the previous slides. And in the top population, we see complete management or clearance of the tumor. This product was directed at a Raji tumor in these mice. And in the bottom panel here, we see failure of the electroporated population to manage that tumor after about, looking on the right-hand side, after about day 12. Although we're showing the highest dose here, we also see at lower doses, we did this at 1, 2, and 4 million cells per mouse, and we see a nice escalation dose dependency when we look at those three dosages going into the animals. And although this result is eye-catching, it was unsurprising to us, having been built on top of an in vitro data set, including cell phenotype, metabolism, avidity, which we did in conjunction with Lumix, RNA sequencing, and then looking at apoptosis and cytotox. So if you like, bringing this together in terms of that complex triple edited product, we demonstrate more potent cytotoxicity in vivo with soluporated cells. So looking to the future and our work ahead, having optimized the transfection manufacturing step, our team had anticipated looking up and downstream of that individual step towards other key steps in the manufacturing process. And here we're looking at a typical autologous T-cell product manufacturing process. So with the same ethos of preserving the cell health and functionality, we've conferred the same thoughtfulness, if you like, into how we expand our cells. And after transfection, we can leave those cells within the same vessel. So you're not transferring them, you're not touching them, you're preserving the process, if you like, and expanding those cells within the vessel. We can also activate the cells, transduce them, concentrate and harvest them as well. So beginning to piece together the full manufacturing process within one single-use disposable, which is available at GMP as well. And we look forward to presenting data on this process put all together end-to-end at upcoming meetings. So in summary, it's an excellent time for people at this meeting to partner with us. We're eager to get our technology into the hands of others and build that experience with the therapy that you're developing, whether that's an iPS therapy, a T-cell therapy, or indeed something else. So with that, I'll conclude. Thank you very much indeed for your attention. Happy to take questions if there are any. Thank you very much.